to the last of the last session of this workshop. We have two talks for you. We'll start with Bernadette Julier, who is not only the coordinator of the eucleg project, but also the alfalfa species leader. Thank you, Juliette.
Okay, thank you, Catherine. So I will give this presentation about genomics assisted breeding in alfalfa. So it's an overview of a work done by a group. So I come back to alfalfa or lucerne. It's a major legume species with the highest protein production per hectare in temperate climates over the legume species. It's quite drought tolerant. It has a convenient protein energy ratio. It provides some advantages on ruminant health, and it has, of course, very positive effects in the rotation. So it's, as already stated, it's an allogamous species, and the varieties are synthetic populations. And in addition of that, it's an autotetraploid species with 32 chromosomes. So this being said, what about traditional breeding methodology? So it's based on phenotypic traits, of course. The first step, as explained by David, is a nursery of space plants, and then a second step is done with a progeny testing. So schematically, you have the introduction of new origins, and you study your breeding pool in a nursery of space plants, where you can choose individual plants on their proper value. So it means on heritable traits. So this space plant nursery is studied for 3 years, and you can study about 5,000 plants or more if you can. Then you harvest seeds on the mother plant, and you test this progeny in microplots for 3 years. And so here you can have from 100 to 200 progeny testing. And depending on the value of the progeny, you can go back to the mother plant and polycross the best plants. So it's another year, an additional year. You can have 20 to 30 polycross a year. And then you study the progeny in a multi-site trials for 3 years, and the best polycross goes to a variety registration test. So these progenies are the basis of a new cycle of the recurrent selection. So this is a theoretical breeding method, and quite often, in fact, the mother plant no more exists when you have the results of the progeny test. So in fact, the polycross of the best plant is based on plants or seeds, usually plants or seeds collected in the progeny test. So then you lose a part of the genetic progress. So where, so what are the strengths and the weaknesses of this methodology? So the strength is that you can score many traits. You can have early selection for heritable traits in the nursery, and for most breeding companies or breeding actors, the staffs are already skilled for this work. The weaknesses is that some traits are scored just only when you have the stress, and this stress does not occur every year. So it means you need tests in control conditions in that cases. You need the main issue is the number of years needed to have a single cycle of selection. So the cost related to this number of years. And also the fixation of positive alleles is slow, especially for a tetraploid species. So where genomics could assist breeding, it could assist breeding in the choice of the origins to be introduced in a breeding scheme. It could help the selection of plants from with the help of genomic selection. And it could also help the choice of plants used in the polycross of the best plants based on the diversity of genetic background. So we will have a discussion about this point. So before going to genomic assisted breeding, we have to develop markers. I will go back to this point. And then I will explain a little bit more about the management of genetic diversity, genome-wide association study, and genomic selection from eucleg results. So about marker development, before eucleg, we had in the past low throughput markers such as SSR and AFLPs with the difficulty of tetraploidy, so it was not easy to have the dosage of the alleles. Then 10K SNP array was developed, but as explained by Leif, it was too expensive, especially when you want to study population in which you have to represent a population by at least 20 or 30 plants. And the array was not that big with 10K SNPs. Then GBS was developed, and it was interesting to see that it works very well, in fact, for a tetraploid species. In most cases, we had less than 40K markers, and in many publications, we have seen quite a huge proportion of missing data, and this is an issue. So in eucleg, we have developed an improved GBS protocol by a study of different restriction enzymes to reduce missing data and to optimize the cost of GBS. So we have tested a number of enzymes or pair of enzymes, and we have sequenced the GBS reads and fragments and done some analysis, map the reads on the reference genome and analyze the number of loci, a load or with resampling of the number of reads. And we have seen that for in two cases, so with PST1 APICA1 in blue or PST1 MEC1 in red, in green, sorry, we have a clear plateau, meaning that with about 10 million reads, we can achieve a stable number of loci. So it means that we have less missing data. And indeed, so we have chosen PST1 MEC1 because it was the pair of enzymes that was already chosen by Red Clover. So it means that given the synteny between the two species, we may be able to compare the markers that can be important for trait variation. So we have chosen this restriction enzymes. We have used a reference genome sequence, a new one delivered by Chen et al. In China, and we have obtained allele frequency of each accession. So at the end, we were able to sequence more than 1,000 accessions. We obtained more than 30,000 loci. And within each loci, we had several SNPs. So we obtained more than 200,000 SNPs with less than 5% of missing data per SNP. So a huge number of SNP with a very low ratio of missing data. And if we choose to add a new threshold, so without any missing data, we have more than 100,000 SNPs. So, and these SNPs are covering very well the genome. So we have this tool available now, and it's a great progress for alfalfa genetics. So about management of the genetic diversity. So before eucleg, we had quite a number of knowledge, of course. We had an overview of world diversity with a center of origin in the Middle East and a trace of introduction in the Western Europe or North Africa. And alfalfa followed migration or invasions with Greeks, with Romans, and with Moors. It hybridized with the subspecies falcata, and then it moved towards Americas and Australia. And there is also a story of introduction towards China about 2,000 years ago. In the past, we also had a quite nice view of diversity with a large among accession diversity, of course, with phenotypic traits and molecular markers, but also a huge within accession diversity, as shown here with phenotypic traits or SSR. So a very large diversity within the varieties with phenotypic traits and even more with markers. With eucleg, we have a revision of diversity overview. We have studied more extensively 400 accessions. So there are land races and cultivars, the dormancy, autumn dormancies from 3 to 7 mostly. Most of the accession comes from Europe, but we have also accession from North and South America, from China, Middle East, and one accession from China. So we have obtained these GBS markers, and we have carried out a PCA on with these markers, and we have obtained 7 groups. You can see here the different groups. So we have a first group represented with only 2 accession close to the subspecies falcata. We have quite a clear different group of accession coming from China. And we have also some groups ranging from Europe to America. So they are connected, more or less close, but at the end, you can see that the group number 3 with accession from France and Northern Europe this group is quite far from another group, the group 2, composed by accession of varieties from North America. So it means that there is a variation and a structure that is not completely far from geographic origin of the varieties. And for people who are not well aware of alfalfa, there is just another point with diversity. We have calculated some FST among groups, and you can see, so not with group 2 composed by two varieties only, but you can see that the FST as a whole are quite low. But the group giving the highest FSTs is a group 6 with the accession of China. For most people in charge of alfalfa breeding, autumn dormancy is a very important trait, and it gives a strong structure to the breeding programs, because usually we are working within autumn dormancy range of variation. And here, if we put a dormancy score on the PCA plot, you can see that the dormancy is not a way of sorting the variety. So the diversity is not linked to the autumn dormancy score. And it's quite a new result, in my opinion. Okay, so here, so you can see that with this project, we have revised the diversity overview. So we have a diversity in China that is quite different from that present in Europe and America. But also the diversity in Europe and America is different. And even if most American accession originate from Europe in the past. And the structure observed is not associated to autumn dormancy. Then I move to genome-wide association study. So briefly, the question is, for each marker, is it associated to trait variation? So here on the left-hand side, a marker that is not associated to the phenotype, and on the right-hand side, a marker that is associated to the trait. So before eucleg, so we had in most cases a low marker density, and but we have shown that the linkage disequilibrium is very short in this species. Here studied at the level of a single gene, but in most cases, linkage disequilibrium over 1,000 base pair is broken, with some exception in this gene. So it means that at this moment we had only a candidate gene approach, not a GWAS approach, but it can work as found here in a constance-like gene that was as a variation for this gene was associated to a phenotypic trait, here stem height. So here with in eucleg, we have found again a very short linkage disequilibrium over the genome, this time not over a single gene. And so it means that here after 500 base pair, there is no more linkage disequilibrium. So a very short linkage disequilibrium as expected in an allogamous species. So it means, in fact that a genome-wide association study is possible because we have many markers, and so the candidate gene approach, of course, is available. But if we go to GWAS, we need many markers over the genome. So we have done phenotyping, of course, during eucleg. So we have studied yield and quality. Quality is protein content, fiber content, and saponins. We have studied 400 accession in 2 locations and 2 years after the establishment year. And in addition, we have studied within the 400 accession, we had 100 accession studied in 3 locations across 2 years. We have also studied germination, disease resistance, drought, and phosphorus tolerance, and the interaction between drought and fusarium. So here we were able in eucleg to detect major QTLs on these accessions, and we have used a multilocus mixed model. And what are the results here? Just as an example for protein content, we were able to identify some QTLs with a strong effect, and about 10 QTLs with a significant p-value. If we look at the specific QTL among these ones for protein content, we were able to see a quite nice explanation of variation here with a variation, an explanation of 0.12%. Oh, so sorry, 12.5% of the variation for protein content. So here, if you, so as a whole, we were able to identify QTLs showing from 10 to 20% of, explaining 10 to 20% of the variation. So let's move to genomic selection. So genomic selection, as explained before, is based on a breeding population that you genotype and phenotype. First, you establish a prediction model. You test it with cross-validation. Then you select, after crossing, some candidates for selection. You genotype these candidates and you apply your prediction model on this genotyping data, and you select the plants you prefer. So before eucleg, some attempts were published on genomic selection. The number of SNPs was not so high, and the number of individuals used for this studies was not high, but colleagues obtained promising results with predictive ability reaching 30% in some cases. So quite promising results. So in eucleg, based on our 400 accessions and more than 200,000 markers, we carried out GBLUP prediction, and we obtained quite a good predicting ability between 0.5 and 0.8 for the p-value shown here for dry matter yield, ADF content, so fiber content, and protein content. It's the predicting ability was even better for protein content than for other traits. Then we integrated QTL information in the prediction model with a QGBLUP, and so on the left-hand side, the GBLUP, on the right-hand side, the QGBLUP, and you can see that we obtain very, very high p-value over 0.8. And this is very, very interesting and promising. So as a consequence in breeding, we have GBS as an efficient technique to reveal markers. So we can have allele usage in individuals, as shown before, but in other studies, I mean, but we can have allele frequencies in population. We have a high coverage of the genome with low missing data. So this will be useful to analyze genetic diversity and genetic determinism of breeding traits. So these markers can be used to manage diversity, genetic diversity. We have some specialization of the breeding pools in EU, in Americas, and in China. So we can use GBS markers to decide on the introduction of new genetic diversity in a breeding pool. We have seen that genomic selection models provide high predictive ability, and these models have a better predicting ability when we are able to include QTL effects. So these models are ready to be used to select promising individuals in breeding pools. What do we still have to do? So probably we have to extend the analysis of alfalfa diversity from dormancy 3 to 7, as done here, to the whole species complex, including probably white populations, if possible. We have to improve cost efficiency of genotyping. This cost was in agreement with our friends for research program. We have to see if we can decrease this cost to be applied in a breeding program. We also have to calculate genetic gain with genomic selection, and of course, it depends also on the cost of genotyping. And then we have to estimate cost efficiency of genomic selection. And of course, we have to implement genomic selection in breeding programs to go from the theory to the practical aspect. Here I propose implementation of genomic selection in breeding programs. So if you consider the introduction of new origin and introduction of and breeding pool, of course. So the breeding program starts with growing seeds in greenhouse, and as soon as possible collect leaflets and have genotyping and prediction, genomic prediction. And at this stage, after a few months, you are able to choose some plants to be established in a polycross with the best plants. Then you have the polycross, and after 1 year, you have seeds to test the polycross in a multi-site trial. And some of these polycross can be candidates for registration. And so you continue the recurrent selection. You can imagine to have more candidates with genotyping, moving from 5,000 plants in a nursery to maybe 15,000 plants, depending on the cost. What would be the strengths of this breeding program? Of course, a reduced field work, a very early selection for all predicted traits, a reduced number of years of the breeding cycle, and a very quick fixation of positive alleles, especially for this autotetraploid species in which you can access to the alleles. The weakness, first, if you have no prediction for some traits, you are not able to select for this trait. And also problem with staff who has to get new skills to adopt this breeding program. So the question is now, what is the genetic gain and what is the cost efficiency of this genomic breeding? Yes, so we have to calculate, yes, of course. So sorry, I was explaining about the implementation of genomic selection, and so the next step will be, of course, to imagine the updating of genomic selection equation, because up to now we have an equation, but how to make it living. So the question is how to use the results of a new progeny studied in new polycross studied in a field. So having phenotypic evaluation to update the existing prediction model. So this has to be built with actors of breeding, alfalfa breeding. Okay, this was my last slide, so I would be happy to answer a question.
Thank you very much, Bernadette. We have a number of questions that have appeared in the questions tab. Firstly, what was the cost per sample in your GBS approach?
If I am correct, it's about 50 euros for one population.
Okay, and does that does that include the DNA extraction and all parts of the process? No.
Yes, everything, but yeah, staff time, I'm afraid.
Sure. And the next question is from Philippe Dufour. How many years of phenotyping does it take to build the genomic selection model and calculate the prediction accuracy?
Well, in our case, we have used data from 2 years of phenotyping in field. So meaning, in fact, 3 years, because we haven't used the first year, the establishment year. So we collected the data over 2 years. So with alfalfa, with 4 to 5 cuts a year. And then we were able to, then it depends on the number of people you can devote to this task for the calculation.
Thank you. And the next question is, how often do you have to renew the prediction model?
Good question. Thank you, Leif, for this question. It's really a question for all the speakers today and other scientists. Well, we don't know, in fact, and well, maybe the first thing is we have to test the efficiency of the quality of the prediction depending on the accession you are studying. So I've shown some groups of accession, some grouping. So we have to test if a prediction model is valid for all types of accessions or not. So this is the first question. And it's related also because once you start using a prediction model, of course, you select plants, so you change the genetic basis of the material. So so of course, we have to check this and to learn from experience, I'm afraid. So we have to, yes. My idea would be that maybe we could use, not start again to zero. So meaning we don't need to collect so many accessions and study them again in field trials, multi-site trials. But maybe we could use this first set of information and then add new information coming from new trials and new accessions, of course. So, but it's not so simple at the mathematical level, and we have also to find an organization to do that.
How much, next question is, how much of the variability for each trait change between the years?
Well, we have some changes in variability, and we have also some interaction between environment and genotype. So here we have tried to predict, in fact, a mean value of the populations. So we have based, so for example, yield is annual yield. And we have, yes, we are able also to predict, for example, we have data from 2 sites. We're able to build equation to predict the traits in each environment. So here we have done the hypothesis that we have a better evaluation if we look at the average value of the populations over all the sites that were available. So yeah, the question of G cross E interaction is of course of major importance.
And the final question that we have is, were the prediction accuracies that you showed cross-validation results?
Yeah, exactly. It was the equation were done on a subset of accession and used to predict another subset. So there was a test set to calculate the p-value.
Okay, thank you. Okay, well, thank you very much, Bernadette.